Can biologists predict what traits of organisms will evolve? You might be surprised. Let's take the case of teeth and toe bones. They might not be glamorous, but they illustrate how knowledge of developmental biology is being used to predict biological evolution. Molar teeth form in a sequence from front to back. The growth of each tooth is inhibited by the preceding tooth in the sequence. Experiments with mice have established that manipulating molecular signals in the first molar changes the size proportions of the entire row of teeth. In short, molar teeth develop as a module. This means, for instance, that knowing the size of two teeth allows the size of a third to be predicted. This allows biologists to develop mathematical models that link levels of gene activation and inhibition to teeth size proportions. Intriguingly, it turns out that these models can successfully predict the number and shape of teeth of not only mice, but of several groups of rodents. In fact, the teeth data for virtually all mammals lie on a single line. Herbivores, who tend to have more even-sized teeth, and carnivores with their less even-sized teeth are all positioned on the same gradient. Rather than being free to evolve any shape or number of teeth, developmental mechanisms have created pathways of selectable variation along which natural selection may act. Selection is actually pushing evolving populations up and down these paths created by development. Historically, such cases have often been understood as developmental constraints, the idea that developmental processes prevent some impossible forms from arising. However, the existence of exceptions to the pattern, rodents such as voles with different ratios of teeth away from the line, demonstrates that this traditional way of thinking isn't quite right. Developmental mechanisms don't just rule out certain impossible body shapes, leaving natural selection free rein to determine tooth shape elsewhere. By biasing the phenotypic variation available for selection, developmental mechanisms part determine which features evolve. Impressed? Well, scientists have actually gone further. They've used the same model of tooth development to predict the development and diversification of other anatomical structures that also arise through sequentially forming segments. For instance, finger and toe bones, known as phalanges, vary widely in number and size proportions across vertebrates. Yet successive bones within a finger or toe exhibit highly predictable relative proportions. They too all lie on the same line. These digits evolved with the dispersal of amphibians onto land around 400 million years ago, and hence the same patterns of toe bone variation are seen in birds, reptiles, amphibians, and ichthyosaurs. In birds, running is associated with unequal length phalanges and perching with more equal-sized toe bones. Raptors have an elongation of the bones at the fingertips, seemingly caused by selection for grasping. Yet in all cases, phalanges have evolved along a highly specific path predicted by the model. These studies of development are exciting, as they're helping to make evolutionary biology a more predictive science. Curious? Check out Evolution Evolving now.